In our last video, we introduced the concept of autism subtypes and why studying well-defined biological subgroups is where autism research is actually making progress. Today, we start with the first subtype, megalencephaly-associated autism. Megalencephaly means abnormally large brain volume, and in a specific subset of autistic individuals, the brain grows measurably larger than typical, particularly in the first 2 years of life, precisely when the most critical period of neural circuit formation is underway. This is not a subtle difference. Studies using MRI have documented brain volume increases of 10% or more in some autistic children during this window. The overgrowth is most pronounced in the frontal and temporal lobes, regions critical for social cognition, language, and executive function. But here is what makes this scientifically interesting. The brain is not just bigger, it is differently connected. Megalencephalic brains show altered patterns of long-range connectivity. The white matter tracts that allow distant brain regions to communicate with each other are disrupted. At the same time, local connectivity, the dense, short-range connections within a region, is often increased. The result is a brain that is, in some ways, overconnected locally and underconnected globally. And this pattern, too much local processing, too little long-range integration, maps remarkably well onto the cognitive profile seen in many autistic individuals. Strong local processing, strong attention to detail, but difficulty integrating information across domains. So what is driving this overgrowth? The primary mechanism involves the mTOR signaling pathway, a cellular growth regulator that controls how fast cells divide, grow, and make proteins. When mTOR is overactive, cells grow too much. When it is properly regulated, growth proceeds normally. In megalencephaly-associated autism, mTOR regulation goes wrong early in brain development, during the prenatal period when neural progenitor cells are dividing and the cortex is being built. Too many neurons are produced. Too many synaptic connections form. And the normal pruning process that refines these connections during development, the process of eliminating excess synapses to sharpen neural circuits, does not work efficiently enough to compensate. The result is a brain with more neurons, more connections, and less efficient pruning than typical. More is not better. Precision is better. Now, why does this matter for autism research beyond this specific subtype? 3 reasons. First, the mTOR pathway is druggable. Compounds that inhibit mTOR, called mTOR inhibitors, the most well-known being rapamycin, already exist and are used clinically for other conditions. Studies in animal models of megalencephaly have shown that mTOR inhibition can partially normalize brain growth and improve behavioral outcomes. Human trials are in early stages. Second, the overgrowth phenotype is measurable. Brain volume can be detected by MRI. Early brain overgrowth in the first 2 years of life could potentially serve as a biomarker, a measurable signal that identifies a specific subtype of autism before behavioral features are fully apparent. This is exactly the kind of early biological marker the field has been searching for. Third, and most importantly, megalencephaly is not an isolated phenomenon. The same mTOR pathway that drives brain overgrowth in this subtype is implicated in PTEN autism and tuberous sclerosis, 2 other subtypes we will cover in this series. Different genetic entry points, same core mechanism. This convergence is telling researchers something important about a shared neurobiological pathway in autism. One important caveat before we close. Not all autistic people have megalencephaly, and not everyone with megalencephaly is autistic. This is a subtype, a specific biological profile found in a subset of autistic individuals, not a universal feature of autism. The value of studying it is not that it explains all autism. It is that it explains some autism precisely. And that precision is what opens the door to targeted understanding and eventually targeted treatment. Follow for more real neuroscience. No tips, no hype, just the science your family deserves to know. Next, microcephaly-associated autism, when brain development is restricted, and what the opposite end of the brain size spectrum reveals about how autism emerges.